Good morning. I'm really conscious of my accent now. Thank you, Ruben. Thank you, Jean. It's always really lovely to be here. Thank you, Joanne, for helping with all the logistics. So I would like to take you all on a journey from the really simple ideas behind why an MPN happens all the way to some of the complex research. There'll be some old stuff here that some of you will be familiar with for those that haven't heard it before, and there'll be some new data as well because it's a fast-moving field. So starting with the basics, on the left we all recognize bags of blood, and on the right is what our blood looks like to a hematologist looking down the microscope. This is just blood from your arm, peripheral blood taken. So I have my cursor here. These things are the red cells, and the reason they have these little white divots in them, I tend to ask prospective medical students who come to St. John's for their University of Cambridge interview why there's a little white divot in them, and I get all sorts of interesting answers. But actually, it's really because they're a disc which is flat in the middle. They're a bit squished in the middle because they're not like a balloon. They're like a squished up balloon. And so because of that, the hemoglobin tends to be around the outside of the red cells, not much in the middle because it's collapsed, and that's why you have this lovely little disc shape. And then these big, sort of slightly scary-looking cells are lymphocytes and neutrophils. They're all different types of white cells that help fight different types of infection. And these tiny little things are the platelets. They're really like crumbs that help make blood clots and help make the scabs. So this is what we are used to and what we're trained to look down the microscope at. And when you look at an MPN blood film, PV-ET doesn't look hugely different. ET will have more platelets. PV doesn't look that different, but myelofibrosis starts to look a little bit different. And basically what we're doing is the peripheral blood is really like the manufactured red cells. And it's all made in the bone marrow here. And this is a picture of the bone marrow. And you can see that the bone marrow looks rather different to the peripheral blood. These big blobs here are just fat cells within and fat spaces within our bone marrow. And we all know that bone marrow is quite fatty, and my time was when we'd give it to children to chew on. And what's happening in the bone marrow is you have stem cells, hematopoietic stem cells that make progenitors, and then they make the white cells, the red cells, and the platelets. And this is a sort of parent cell of a platelet called a megakaryocyte, and it's from this that those crumbs of platelets get thrown off. So those are really bizarre cells, really large ones. And what happens in myelofibrosis is you see more of these cells out in the peripheral blood. But really, apart from that, the peripheral blood in the previous picture doesn't look that different in MPNs. And so what's happening in myeloproliferative neoplasms really is that there are no abnormal cells present in the blood or the peripheral blood of the bone marrow. What you're seeing is just more overactivation of the system. So whereas these blood stem cells make leukocytes, which is a term for white cells, red cells, and platelets, in myeloproliferative neoplasms, it's just an overdrive. And it tends to be an overdrive in this section here. The white cell count can go up in some individuals, but it tends to be either the red cell count that goes up in PV or the platelet count that tends to go up in ET. And then, myelofibrosis can be a bit of a mixed picture. So why is that? Well, before we go there, these are really ancient conditions. It's not a modern era phenomenon. For hundreds of years, really, we've known about MPNs in humans. And MPNs also occur in other species. So really what that's telling us is that there is a very ancient evolutionary history behind multiple sort of species developing this overactivation of what is essentially normal blood cell production. And in this talk, I'm going to go through some of these questions. You've got PV at the top here, and actually what this is showing is that if you take blood from your body, just normal blood tube, normally the nurse or the specialist will take some blood and it gets whisked away to the lab. But imagine it got forgotten and got left on the shelf. It separates into plasma and the red cells here at the bottom. And who's familiar with their hematocrit? Hands up if you know your hematocrit. Your hematocrit is the percentage of this versus that. So it should be, you can see it's hard to tell because of the shape, but it should be less than 50%, less than 45% ideally in PV. And really what you're looking at is if you let it sink to the bottom, what proportion of your entire volume would it take? And this obviously is in PV. So this happens to be my old boss's blood. who used to come here to do the conference before he passed the baton to me, Tony Green. And then on the left here is an individual with PV. Obviously, you can see at diagnosis, there's a lot more red cells than there ought to be once it's sunk to the bottom. ET here, you can all now diagnose this as hematologists, lots of extra little platelets. And this is actually a picture of the bone marrow, not the blood, in myelofibrosis. And the black lines are the scar tissue, the collagen that forms in myelofibrosis. So every time I come, I add more questions. So I have 5 questions to get through this time. What causes it? Was it just bad luck? What causes the differences in diseases between individuals? Why do some people get PV, others get ET? How long have I had it for, and how fast did it grow? Just some questions we've come to understand better in recent years. So what causes it? So I love this slide here. Some of you may have seen it before. So on the left-hand side is the first ever time in 1955 where these guys here were able to count the number of chromosomes we have. And what they discovered is that if you take a cell and put it in a sort of salty, low-salt solution, the nucleus sort of swells up, and within the nucleus, which is, so our body has maybe 15 trillion cells, and within each cell is a nucleus, and within each nucleus we contain the code for making us. And that code is in our chromosomes. We didn't know how many chromosomes we had, and this is the first time that it got split up and you can actually count the chromosomes. And the reasons they're different sizes is because each chromosome number is a different size, and chromosome 1 was the longest one. I reckon that's this one here. And then as you go down to 23, you go smaller and smaller and smaller. And they were literally ordered by size and named 1 to 23. And then what happens when you unravel one of these chromosomes here, so one of these copies will be from your mum and one of these copies will be from your dad. So each chromosome, you'll have a mum copy and a dad copy. And when you unravel it and unravel it and unravel it and unravel it, you then get to the DNA here. And the DNA here, so chromosomes really are a way of packaging the most vast amount, 3 billion of these little bases, which is the code for DNA, into a little nucleus inside a little cell, and we have that trillions of times in our body. So I just think it's fascinating, as you can tell. So, so this is now, we've unraveled our genome, we're at the level of the bases, and what's in this? We have things called genes. Genes are basically the code to make a protein, and protein is basically, like, think of it like this. Protein is the cake and the genes are the recipe. So we make these proteins, and JAK2, this is an interesting protein. So JAK2 basically signals. It takes messages from outside the cell and goes into the nucleus and tells the cell what to do. And it's particularly active in blood cells. And there are different domains that people who study proteins have worked out. And this is the domain that does all the action. It's the one that switches on. And this domain here switches that off. And what the JAK2 mutation does is it stops the off domain from working. So if you stop the off domain from working, JAK2 is now permanently switched on. So when you have a V617F mutation, what that's doing is it's keeping JAK2 permanently on. And JAK2 signals from this little receptor here into the nucleus to say make more red cells or make more blood cells in general or more platelets. And normally it switches on and off depending on as you need it. You know, Tour de France runners who have occasionally used a little bit of additional EPO to boost their red cells, JAK2 is taking the signal down to the nucleus. So this is why when JAK2 is overactivated, you get more blood cells. And we know that, I remember qualifying as a doctor in 2004, and Tony Green was at Cambridge then, and him and many other groups around the world discovered JAK2 mutations at that time. And I remember thinking, what's going on? Why is everyone talking about JAK? I thought it was J-A-C-K. It was someone famous. And everyone was talking about JAK in MPNs. And what you realize then, just as I was going through my early training, was that within 5 years, we went from discovery as a world to JAK inhibitors in the clinic, in trials. And I thought, wow, that's amazing innovation that takes patient participation, funding organizations to get behind it, and a whole community of researchers and drug companies and clinical trialists to go from a discovery to something that could benefit patients. And I really valued that move in the MPN field and many other blood cancers for that matter. But what happened was we realized that whilst JAK2 was present in 99% of patients who had PV, really quite common, it was only present in half of ET-MF. Only 50% of ET and MF had a JAK2 mutation. We discovered, not us, but in general, the sort of royal we, discovered that there were some MPL mutations. MPL is a signaler for making platelets, so you can understand why a switch on for that will make more platelets. So MPL mutations are rare. I'd say maybe 1 in 50 individuals with MPN have a MPL mutation, but it essentially does a similar thing, but specifically just makes the body make more platelets. And then there were some other rare mutations, LINC, Sybil. So basically, these are other proteins that switch off the system. And if those got mutated and stopped working, then again, you were overactive, made more. So there were all things that essentially led to the same outcome, but different routes. But again, Sybil and LINC mutations were found in a few % of patients. It didn't account for half of individuals. So I got landed with a project where we looked at genetic sequencing for the first time in MPN patients, and the technology was booming at that time. We're talking about 10, 15 years ago, where suddenly you could sequence not the genome back then, we're not talking all 3 billion bases within that DNA. The technology was still not there, but it is there now. You could probably do that for $100 to $1,000 now, but back then you couldn't. But what you could do is sequence all the genes. So not all the junk DNA in between, but you could sequence all the genes. And that was called an exome. So we had a project where we collected all these samples from the clinics in the UK, and we studied these exomes looking for something that might be commonly mutated in patients with ET and MF. We didn't find it for ages, and there are complex reasons why we didn't find it. I don't know why the slides keep going back. I think it's me, apologies. But eventually, we realized that there were CALR mutations in 2 individuals in our cohort. And then when I went back and looked at the rest of the data, I realized the CALR mutation was present in nearly everyone that didn't have a JAK2 or a MPL mutation. But for reasons in the genetic sequencing, the algorithms had sort of thrown out the data. So just because I had to talk about this last year, Naveen and Ruben asked me to go back and talk about how it all happened. I started digging through emails, and this is actually the very first email on the 13th of July 2013, where I emailed my other boss, Peter Campbell. He's notorious for never replying to your emails, even particularly if you're a PhD student. It's like sending emails into the ether. And I sent him an email going, oh my God, this is exciting, or it maybe isn't. I don't know. I hope it be. And this is the first time I didn't really know how you talk about CALR, because the closest thing on Google at the time was Kal-El from Superman. So I was like, is it CALR? Is it CALR? There was another name, CRT. I wasn't quite sure. So we were like, yeah, well, let's call it CALR. And those are the 2 patients that we first found them in. And there were deletions, which is where you lose a bit of the DNA. And anyway, lots of things. And he wrote back. It's the only time he's responded to my email. He wrote back going, wow, meet me in my office Monday morning. And Tony, who probably didn't have much faith in me as a PhD student at the time, but I think it changed after this, he was in Cornwall on holiday, and he wrote to me going, camp outside Peter's office, call me if he thinks it's real. And I'm like, I think it's real. So anyway, Tony's actually retiring this year, and Peter's also moved on to other adventures. So I'm sort of keeping the genomics flag flying for MPNs. And so this is the new test now. Because there's a deletion in CALR, CALR becomes smaller. And so this is normal CALR, and this is the DNA size, and it sits around here. And depending on what sort of deletion or change or insertion of DNA you have in CALR, it becomes a different size. So this is not the test we use now, because things have moved on, but back then this was the test that we used. So we ran the sample for a JAK2 mutation. If it was negative, we ran it for size, specifically for CALR, and if it was a different size, you knew the patient was positive. And of course, we're not involved in developing drugs and targeted treatments, but the whole world has, deciphered how CALR works, how it actually switches on blood cell production. And essentially what it does, it's really fascinating. What it does is it basically, this little bit in the middle, it attaches onto the top of this receptor and links them together and switches the receptor on. So it does the same thing really as a MPL mutation, which is why CALR mutations will never cause PV, doesn't cause increased red cells, it only causes increased platelets or myelofibrosis. Anyway, back to, so there, it's really exciting at the moment. There are 2 phase 1 clinical trials, CALR antibodies. Lots of the faculty that are going to be here today and tomorrow have personal experience with recruiting patients into these trials. We're not a phase 1 trial center in Cambridge, but it's really interesting to be here and speak to colleagues who are really immersed in all of these clinical trials, including the CALR antibody trials, and watch the space. I think the next 5 years will be super exciting in this space for CALR therapeutics. And there are all sorts of molecules in the CALR space, not just antibodies, but CAR T and all sorts of things that are in development. And I think this sort of AI era that we're all entering in is going to accelerate that for all MPL, JAK2, CALR antibody-mediated drug discovery. So very, very exciting. And 11 years since CALR, I now am a fully fledged researcher. I still treat MPNs, my group's growing, and this little girl in the middle here, the little baby with the ducky, is now 11 years old. So there you go. And they're actually here with us in Phoenix because we're going to take them on a little holiday after tomorrow. So basically, I hope I've explained how changes in genes, JAK2, CALR, and MPL, drive MPNs. Okay, and this is just one little graph here to show how CALR sits at the top, MPL is a receptor, JAK2 sits inside the cell, and they all kind of do the same thing. They just switch it on, and you go into the cell, and these little words here are basically pathways that then switch on blood cell production. So most patients, 90% of patients, have one of these 3. I will touch a bit about the odd patient who might not have these 3, and you may be told you're triple negative, and I can touch on that. Many patients, particularly as we all get older, have additional mutations in other genes. Now, this happens normally as we get older, regardless of whether you have an MPN or not. These are the same genes that we all develop mutations in. It's just part of aging, but sometimes they happen within the MPN clone. So there's a JAK2 mutation or a CALR mutation already there, and then you can have a second event or a third event, and that can lead to the clone growing a bit faster. So that can be common in myelofibrosis. It can be common in advanced PV and ET as well. But it's really something that you see more frequently as you get older. So was it just bad luck? Was it just that I happened to get a CALR, MPL, or JAK2 mutation, and that's why I got the disease? Well, that's what we always thought. But actually, there was this really interesting study published in Europe by the Danish colleagues who started to study whether a JAK2 mutation was present in healthy individuals. And I don't know if any of you are familiar with the concept of VAF or allele fraction. So basically, sometimes we put a number, not always, because we don't really know how to use it clinically, but sometimes we put a number by the mutation. So we might say 10% JAK2 or 90% JAK2 or 20% CALR. It's not important that we necessarily do that. I mean, the time will tell whether that is helpful or not, but that's basically saying of all the blood cells that you make, what fraction are positive. Okay, so it gives you a sense of the amount of MPN that you have. And they did this study where they found that 3% of individuals, so I'd say 1 in 30 individuals, has a 1% JAK2 clone. These are not patients with an MPN. So JAK2 is common in the healthy population. So then you think, oh, okay, why did they have a 1% JAK2 not get a disease, whereas I maybe have slightly higher and I have a disease? Why? And we think at least part of that is germline genetics, and that basically means the DNA that you're born with. So these mutations so far that I've been talking about, these aren't things that we're born with, they're things that just happen in our cells one day as the cell is dividing, a mistake. But actually, there's a lot of DNA that we're born with from our parents, and that's why we all look different to one another, and some are tall and some are short, and some families will be predisposed to diabetes, others will be predisposed to arthritis. And there are various parts of our DNA that we now know predispose to getting an MPN. So it doesn't mean it predisposes you to getting a JAK2, because I think JAK2 mutation is quite common, but I think it's more like fertile ground for the growth of JAK2, which might mean that if it occurs in one individual, it doesn't really grow, but in another individual, if it happens at the right time in the right cell, then it will sort of start to grow, and then that growth can cause a disease. So these are just listed a couple of things. There's something called 46.1. It's a part of our genome, quite common, and it increases your risk of an MPN by 50%. But what we're beginning to realize, the third line down, it say DNA sites influencing normal blood counts, also important. So we did a study last year where we just looked to try and understand what are the sites in our DNA that influence our blood counts. Nothing to do with MPNs, because all our blood counts, if we measured our blood counts, they'd all be slightly different. Our hemoglobins wouldn't be the same. Our platelet counts wouldn't be the same. Some people would have a platelet count twice that of the other, but they're still within the normal range. So what governs that? We think maybe up to 30% of where our blood counts are is down to our DNA. And so we then went and asked in PV and ET, how much do your baseline inherent DNA, how does that influence whether you get PV or ET once you have a JAK2 mutation? And we found that we could predict whether an individual was going to get PV or ET in the context of a JAK2 mutation. And it was really ended up being quite simple. If an individual is more predisposed to having higher hemoglobin, higher hematocrit, lower platelet counts, when they get a JAK2 mutation, they're more likely to develop PV. And similarly, if someone has a germline or a predisposition to lower red cell features but higher platelet features, then in the context of a JAK2 mutation, they go down the route of ET. And similarly, if you're likely to have many blood cell markers, low platelets, low hemoglobin, low white cell count, you're slightly accelerated towards getting myelofibrosis, because your blood counts, you start to get low blood counts. So we found that actually your germline genetics are probably really important in what sort of disease you're likely to get. And then there'll be other talks during this conference that say it might be other things as well, inflammation, and there are, other things that perhaps during our environment will influence how quickly some of these clones grow. So what causes the differences in disease between individuals? I've already touched on the fact that our germline genetics might be important, but these mutations can also be important. So for example, I've told you that a CALR mutation will not cause PV because it doesn't activate that pathway, but a JAK2 mutation can cause all the different diseases because it's a signaler for all the pathways that make blood cells. But we know that a higher level of JAK2, like 90% JAK2 versus 5% JAK2, is more likely to cause PV. And we think it's because to get red cell production, you need two JAK2 molecules down this pathway here to stimulate red cells, as opposed to just one abnormal molecule. So here we go, double copies of JAK2. So sometimes people have a JAK2 mutation, but remember you have two copies of all your genes, one from mum and one from dad. Often, really often, that abnormal copy that you're seeing here, this is the JAK2, it duplicates itself. So what happens is, you've got a JAK2 mutation on the dad's copy of the chromosome, there's mum's copy there, that's fine. The bit that's abnormal doubles itself. So it now affects both chromosomes. You get two copies. And that then drives red cells. So these are little red cells growing in a dish. And you find that it drives the red cell production over platelet production. There's a special type of mutation in JAK2 called the exon 12. It's very rare. Maybe 1 in 100 PV patients will have an exon 12 mutation. And if you have that, it makes the JAK2 very sort of strong for red cell signaling. So people with an exon 12 mutation and JAK2, they don't tend to have ET. It's all about making more red cells. And lastly, I mentioned that there are all these additional mutations that you can get, and it's normal. We're all getting them in all of our blood cells as we get older. If one of those cells already has one of those mutations, and then JAK2 comes along, you tend to have more ET. Whereas if JAK2 came first and then one of these mutations came second, it boosts the JAK2 clone, the JAK2 clone becomes quite big, you tend to get PV. So it's really an interaction of multiple things: the DNA that you're born with, the environment that those cells may have been exposed to, some of the stresses, but then all the genetics within the actual clone itself that's different in different individuals that ultimately shapes the flavor of disease. And, other things. Males, those over the age of 60, higher odds for PV. And then these are all these little sites in your DNA that make you more likely to have ET. And it's the same through for myelofibrosis. Again, lots of mutations, lots of sites that predispose you to one versus the other. So we now have this personalised website here, blood.predict.nhs.uk. The NHS, which is the National Health Service in the UK, we're hosting it, but apparently their digital area is now being disbanded, so we need to move this somewhere else. But it's basically a site where clinicians, it's predominantly for use with your clinician. You can put in the age, the blood counts, everything, your genetics, if there is genetics that has been done. Most patients will have basic genetics done. Some centres, depending on whether there is a clinical need for it, will do more extended mutation analysis. And then it tells you whether the individual is at risk of myelofibrosis or acute myeloid leukemia and various other things. And it's now been certified as a software because we need to make sure it does what it says on the tin. And it's called Predict Blood. But I'm sure now, as we're entering this sort of AI era, there'll be even better models. I was only contacted yesterday by a group in Israel who are developing a machine learning model for prognosis and MPNs, and they're going to try and integrate all the existing models into their model and create some big, it sounds really exciting. So I think watch this space. This feels a little old school now, but it still does the job, and there are many other prognosis predictors out there, like the IPSS, the DIPS score, things like that. But I think in the future, really, we'll be seeing a new era of understanding and being able to predict personally what happens to each individual patient. So how long have I had it for? So I just want to take a moment to explain why some of these mutations occur. I've already talked about how we have trillions of cells. If you think about how many cells we make in our body a day, really there's 3 tissues that contribute to the cells that we make every day. There's our skin. We make a lot of skin cells every day. We're renewing our gut. There's a lot of new gut cells being made every day. And we're making a lot of blood. Of those, the rest of the tissues, they're great. They do their thing. But they don't make lots of cells. Like the nerves, we don't make more neurons every day, but some of our tissues can regenerate. So of blood, skin, and gut, which one do you think makes the most cells every day? Blood, absolutely. So if, let's say 100% of the cells we make every day, 98% of them are blood cells. So as a human, we have to make the most blood cells every day. We make 200 billion red cells every day. We make 10 billion white cells every day. Every time one cell is made, the parent cell has to divide into 2 cells. Every cell division requires a copy of the entire DNA in that cell to make 2 copies, and they have to copy 3 billion bases of DNA. So they're having to copy 3 billion bases of DNA to make 1 cell, and they have to make 210 billion cells a day. So there's a lot of DNA copying going on. And mistakes happen. Okay, so remarkably, not many mistakes happen when we're making billions and billions of bases of DNA a day in our bone marrow. Within every stem cell in our blood, we think, and we have pretty good evidence now, that we develop about 17 mutations per year. And that's 17 over a denominator of 3 billion. So that's pretty good. DNA copying is really accurate. But there's a tiny amount of mutations that we get. And when you scale that up to the number of stem cells that we have in our bone marrow, and we think we have as adults 100,000 stem cells. And that 100,000 stem cells are responsible for making the billions of cells we need every day. And the good thing about stem cells is they can make more cells but still stay as a stem cell. And the new cells go off, do their job, die off, and the stem cells make more. And we think that within our blood and our 100,000 stem cells, we're developing a mutation that affects a protein every 15 minutes. and we have several hundred genes. So that's really telling us that I think this is intentional. I think our stem cells develop mutations because those mutations allow the stem cells to be slightly different from one another, which then mean that they can adapt to different challenges. And actually, all the tissues in our body develop mutations over time, and it leads to a diversity of cells, so that when you're younger, when there's infection, when there's an injury, you have some cells that can cope better than others. So I think it is adaptive. The only cells in our body that really develop the fewest number of mutations are the sperm and the eggs, because they're responsible for taking the human genome and passing it to the next generation. So they don't want any mutations. They want to keep a clean copy. But the rest of the organs, they're fiddling around with having these mutations. And very occasionally, some of these mutations give the cell an advantage, cause that cell to grow, and can occasionally cause disease or a cancer. So it is important that our stem cells are developing mutations, not too many, but not too few, but occasionally this is a byproduct of what is an adaptive evolutionary mechanism. But what this has allowed us to do is, because our stem cells are dividing and making mutations and having mutations every year, when we then look in a cell, we can work out when in time that mutation occurred, because the number of mutations that are in our cells gives away our age. So because you're getting 17 mutations per year, at age 5, at age 10, at age 20, you know how many mutations there are, 17 times how old you are. So when you plot the number of mutations in stem cells, it follows age beautifully. Nobody can lie about their age anymore. We just need to sequence DNA. In fact, we had one individual that we thought was 71, and they had the mutations of a 36-year-old. And we were like, wow, what have they been having for breakfast? And it turned out that it was just an error in our data sheet, and they were actually 35. So it's really accurate. But it allows us to time mutations. It ends up being a DNA clock. The DNA serves as a clock now for us. And what we can then do in this study, which I'll go through briefly, sorry, the text has gone a bit funny, but we took 9-10 individuals with MPNs diagnosed from age 20 to 70 to 80. So we looked at young and old individuals with MPNs, ET, PV, and MF, and we took blood from their arm. We grew it into little colonies in a dish like this. These are a clump of red cells, and we did whole genome sequencing, because CalR detection and discovery was 10 years ago, but now whole genome sequencing is like buying a pair of socks. It's really easy. Suddenly, I mean, I work at a genomics center, so there, if you want to do whole genome sequencing, it's great. If you want to do something less, they're like, why would you want to do that? So whole genome sequencing is really accessible now and cheap, certainly within research setting, not clinically, but research setting. And what we did is we did whole genome sequencing, not just of the individual, but we did it of the individual cell, because we could do that. So when you do whole genome sequencing of the individual cell, you then get all the mutations that that cell has acquired in its lifetime and its parent's lifetime, all the way to the start of that human body, the fertilized egg, if that makes sense. So imagine we take all the individuals in this room and we draw a family tree of how we're related. How far back do you have to go to find that we had a common ancestor? We're essentially doing that within the cells of the MPN to see how are they related to one another. And that allows us to trace back the growth of the tumor. So what we're doing essentially is using mutations now to build a family tree. And I won't go into how we do that, but this is a family tree of an individual diagnosed with ET at the age of 21. So every little dot at the bottom here is one of those little clumps of red cells that we did whole genome sequencing of. And the line going from this one all the way up and then through this family tree right up to the middle here tells us the parent and the parent and the parent and the parent of that cell all the way to the fertilized egg and what mutations have been passed on. So we're not talking inheritance of mutations, but within a tissue. So we're not talking about the parents of this individual, we're talking about the parents of the cell, the stem cells. And here, these little red cells here have a JAK2 mutation, and these red cells here don't. So this is normal blood. This is just normal, what we call wild type. It's a great name, right? Your wild type hematopoiesis is your normal blood. And then this is the MPN clone with the JAK2 mutation. And you can see that at this point here, the JAK2 mutation was acquired and then it started to grow. Rather than just being one little line down here, like all of these, it's kind of gone boom and burst into a clonal outgrowth. And this branch here was acquired before 80 mutations of life, and it sort of tells us that the mutation occurred between 6 weeks post-conception of 1.3 years of age. And this individual was diagnosed at age 21 just with a high platelet count. They were actually quite well, just platelet count was sort of quite high. And you know, as you get older, these branches become longer and longer and longer because I said, right, you're getting 17 mutations extra every year. So the trees get longer and longer and longer, and you can work out where things have happened. And we have loads of these trees now, and you can see a second mutation coming, a third mutation coming, and you can really find the entire history of the tumor. And what's interesting in MPNs is it's always occurred early, and it's always grown slowly, super, super slowly. And this is another individual. These were twins, actually, twin A, twin B, both diagnosed with CALR-mutated MF within about 2 years of each other. These were not our individual patients, from a family in Oxford, from some collaborators. And when they did a similar study, but not using whole genome sequencing, using other technology, they were able to time that the CALR event occurred in utero before. Well, they think that it occurred after the clump of cells split into two twins, but then often twins will share a bit of blood, particularly if they have the same placenta. And so what probably happened is it occurred in one twin, got shared with the other twin, became stem cells in both twins, and then 38 years later, they developed MF within a year or two of each other. So this, this was a really fascinating study that showed that really many of these mutations occur really early. It's important that they occur early because they grow slowly. If they occur later, one will never get disease because the clone won't even be 1% by age 100. So we're biased towards finding individuals or patients where the mutations occurred early because that's why they have disease. There was enough time for it to grow. And actually, I don't know, I'm sure in the UK we do heel pricks in newborn babies to test for cystic fibrosis and other bits and pieces. And the JAK2 mutation, indeed, in Oxford, they did they went to this sort of old closet of super old heel pricks from like 40, 50 years ago, and they found the JAK2 mutation in several individuals within the heel prick. And this is in line with the fact that the JAK2 mutation is found in the healthy population. So we're getting the sense now, it's a question of when it occurs. If it occurs early enough, it has a chance to grow, and how fast it grows. Sometimes JAK2 occurs second. So these are some other pictures of trees where you can see lots of different additional events have occurred within the MPN clone. I won't go into it, but it's helpful for us to understand this scientifically. So how fast did it grow? We were able to deduce that in two ways. We took serial blood samples from these individuals and tracked the clones physically, how big were they? But we also looked at the pattern of the tree. I talked about how you have this little branching pattern as the clones grow. If that happens really quickly, it means the clone was growing quickly, and if it happens super slowly, it means the growth rate was low. So we had two complementary methods, and we were able to time how fast these clones grew. So these are individuals with clones just one mutation, then you have two mutations, some individuals had 3, 4, 5 mutations. And what you see really is that you have some individuals that have, I call them the slow burners. They have one driver mutation, usually occurred early, took decades to expand, and they often present in mid to late life. Then you have the fast growers. The fast growers tend to be the clone with multiple driver mutations, and they tend to be more in myelofibrosis, where you've had time and decades to get a second event or a third event, and they tend to grow fast. But also, you have some individuals here, like this individual just has a JAK2 mutation, and they were growing at about 70% per year. That's like an interest rate on a bank account, basically. How much is it growing? And that individual was quite fast. 70% per year growth is quite high. And they were the individual that presented at age 21. So maybe that's why they presented at 20 and not 60, because their clone was growing at 70% per year and not 20% per year. And it may be that those individuals in the community that don't have an MPN but have these mutations, they're maybe growing at 5% per year or 3% a year, which means they'll never get disease because it's just, simple maths. If you think about compound interest on a bank account, there's a huge difference in 3% versus 15% if you look over the course of 50 years. And so what we're really talking about is when does it occur? What is that interest rate on that clone, which is determined by all sorts of factors, the DNA that's within your body, the environment those cells are in, as well as what other mutations might be present. And then that ultimately determines how many cells you have and then, what problems will be occurring as a result of those abnormal cells. So overall, I hope I've explained to you today a little genomics journey that, blood cancers, including MPNs, are driven by acquired DNA mutations in our blood stem cells. There's variable speeds of growth of these abnormal clones, slow to fast in different cancers. MPNs in general are some of the slowest growing cancers, even for blood cancers. Mutations can be acquired very early indeed, but that is not a cause for worry. It just makes sense given that they're so slow. And it doesn't mean that the mutations were missed, because I don't think we would be benefiting anyone from finding these mutations earlier, because we'd find them in everyone. I think what's more important is finding it in individuals where it's at risk of growing fast. And that's really our aim for the future, is if we are going to start trying to detect these mutations earlier, really we need to differentiate between people where it's growing fast versus people where it's not going to cause a problem. And then if we can understand more carefully what influences these growth rates and then slowing down the growth rates, then we could potentially enable preventative strategies in the future. But on that last bit, we're not there yet, but there's lots of efforts. And in some countries in Europe, they're screening the healthy population for JAK2 mutations and then trialing early safe treatments to see if we can reduce clone size, but we don't know the outcomes of those yet. So happy to take any questions. Thank you very much.